Moving around seems like one of the most simple and intuitive things that we do every day. We move around most of the time without even thinking about it. But under all that movement, there's a lot of complex microscopic machinery going on. And in today's video, I want to go over that machinery. So we all know that movement is caused by our muscles. Without our muscles, we wouldn't be able to move at all. But in today's video, I want to go a little bit deeper into what kinds of muscles we have and the different parts of a muscle fiber. But adding on to that, we also know that our nervous system is involved in helping us move around. So I also want to go over the nerves and the neurons involved in this movement. And after talking about the macroscopic scale of things, I want to go down to the microscopic level and talk about muscle contraction and the little golf clubs that we have in our muscles, and also the sliding filament theory. And after going through all the details, I want to talk about how these actually apply to everyday life, including exercise fatigue, medical conditions, and even conditions in dead people. And now with our agenda out of the way, let's get into some foundational concepts. So what kinds of muscles do we have? We have three types of muscles. We have skeletal, cardiac, and smooth muscle. So when we're looking at the muscles and we're trying to remember what they do, we're going to look at their names. Skeletal muscle is going to be connected to the skeleton. Cardiac muscle is going to be the heart muscles. And smooth muscle is going to be the digestive tract muscles. So anything in the stomach, mouth, or esophagus. So I could easily make videos on each of these types of muscle, but today's video is going to be focused on the skeletal muscle. So these are the muscles that we think of most of the time when we're talking about muscles. We're thinking of the bicep, we're thinking of the quads, and we're thinking of the triceps. Muscles that we can voluntarily control. Firstly, we're going to look at the different parts of a muscle. Okay, so firstly, we're going to start with the whole muscle. This is what we see in all the diagrams. This is what we see in all the anatomy classes. So if we go a little deeper, we see the fascicles, which are bundles of muscle fibers. Now, an interesting way to remember the fascicles are bundles of muscle fibers is by remembering the word fascist. If you've ever heard the word fascist, then you may have seen that a symbol that used to be used as a fascist symbol is a bundle of sticks tied together. This word comes from the Italian root word, which is fascio, which basically just means a bundle. So if you ever want to remember what a fascicle is, think about this symbol that was used by the fascists. So now if we go a little deeper, we see the muscle fibers, which are the muscle cells. And then if we continue on, we see the myofibrils, and then we see the sarcomeres, and then the myofilaments. Now, if you've noticed, the structure is very much like a rope. There's a bunch of smaller threads threaded together into a big mass of muscle. So now the part that's most critical, most important for us is the sarcomere. The sarcomere is the most basic contractile unit. If you're doing any kind of anatomy class or biology class, they're going to ask you, what's the most basic contractile unit of a muscle? It's going to be sarcomere. Now, when we're looking at the sarcomere, we have the Z lines, the A band, the I band, and the H zone. Now these are just different zones designated on the sarcomere that contain different things. And then along with that, the thick filaments are called myosin, and the thin filaments are called actin, troponin, and tropomyosin. These three are all put together and are classified as the thin filaments. The thin filaments are going to be anchored to the Z lines and extend towards the center. The thick filaments, that are the myosin filaments, sit in the middle and are anchored to the center or the M line. So I think as we're starting to see, these labels, the I band, the A band, the M line, these are all just labels of where these filaments are located and how much of these filaments are located there. So for example, the I band only contains thin filaments. It's going to be only the actin. There's no overlap of the thin filaments. An easy way to remember that this is just going to have thin filaments is, remember I as for isolated thin filaments. It's only going to have thin filaments in that area. The A band, the darker band, is going to span the entire length of the myosin filaments, and it's going to include areas where the thin and thick filaments overlap. This part is going to stay the same during contraction, as we're going to see later. Now inside the A band, there's going to be a much lighter H zone, which is going to contain the thick filaments only. There's going to be no thin filaments here at all. Now this is only in the relaxed state of the H zone. This is not always going to have no thin filaments, because when it's contracted, thin and thick filaments are going to overlap over one another, and there's going to be an overlap at the H zone. The M line is probably the most easy to remember. It's just the central line that's in the middle of the sarcomere, and it's going to be anchoring the thick filaments. So I'm sure we've all heard that we need protein to build muscles. There's the crazy protein craze right now where every single food you see on the shelf itself has some protein. Now the reason that is because these sarcomeres are made of basically entirely protein. There's going to be just a bunch of protein types in here. Now we've been talking about the thin and thick filaments, but let's talk about the thin filaments first. The actin is a long helical chain with myosin binding sites. So it's basically just going to be there for myosin to bind to be able to move it. It's going to be very thin, and it's going to have the troponin and the tropomyosin on it. The thick filaments is the myosin. This is the golf clubs I was talking about in the intro. These proteins use ATP, and they are able to get into a cocked position and move the actin in order to allow for contraction of the sarcomere. Now the troponin and the tropomyosin are just regulatory proteins. They're basically just the police around here to just make sure that the myosin and the actin are binding when they're supposed to be binding. So the tropomyosin just blocks the binding sites on the relaxed troponin, and then when calcium arrives and binds, the troponin will move and we'll be able to have their binding sites open for myosin to bind to. Okay, so that's all the parts of the sarcomere, but in order to understand how it really contracts, we need to go all the way back to the motor unit. Okay, so what is a motor unit? A motor unit is made up of two things: a motor neuron and a bunch of muscle fibers. A motor neuron is a neuron that's in the spinal cord that stretches out and innervates a bunch of muscle fibers. So we have different sizes of motor units. Motor units can be very large or they can be very small. The small motor units are used for very precise movements. So if you're drawing something, or if a surgeon is doing a surgery and they need to be very precise with the movements they do, they're using their small motor units. If you're doing the activities that require a lot of muscle movement, a lot of muscle strength, you're going to be using larger motor units, which is basically your motor unit recruiting more muscle fibers in order to be able to hold up the force that you're putting upon them. So you're going to be using a lot more larger motor units, for example, if you're doing a push-up compared to when you're writing something on paper. One of the key terms that we need to remember for this is motor unit recruitment. This is basically when there's more force applied, when there's more force needed, the motor neuron is going to recruit more muscle fibers in order to do the job. This is basically the equivalent of the military draft for your muscle contractions. Now, I'm not going to go too in-depth into neurons and how they work in this video, but for the purpose of understanding the contractions and how the signals are delivered, know that there's an action potential that's delivered to the axon terminal. And then after it's delivered to this axon terminal, the axon terminal activates and knows to release chemical messengers. Okay, so now let's put this whole thing into context. Let's say you're doing some bicep curls. The bicep curls and your decision to do the bicep curls is going to be the sensory input that's going to go to your brain and decide that, okay, I'm going to move my muscle. Now this sensory input from your motor neuron is going to go down to the axon terminal that connects to the muscle fiber, or the sarcolemma. Now the place where this neuron meets the sarcolemma is called the neuromuscular junction, or the motor end plate. The motor end plate is on the sarcolemma, and that's what receives the chemical messengers. So once this action potential arrives at the neuromuscular junction and at the axon terminal, which is the end of the axon where the chemical messengers are stored, a bunch of calcium starts entering into the neuron terminal. And then acetylcholine is going to get released out of vesicles from this neuron. It's going to get released into the space called the synaptic cleft. This is just a little tiny area where the chemical messengers can flow through and go bind to the receptors at the motor end plate on the sarcolemma. And the sarcolemma is basically just the membrane of the sarcomere. Anytime you hear lemma, it's going to be talking about membrane. Okay, so now we have the acetylcholine binded to the receptors on the motor end plate. This is going to signal the sarcolemma to open its sodium channels and bring in a bunch of sodium, changing the polarization of the membrane. This is going to send down an action potential that goes all the way down the sarcolemma to the T-tubules. So what are these T-tubules that I'm talking about? T-tubules are basically just the membrane that goes down deeper into the fiber. This membrane carries down the action potential deeper into the fiber to the sarcoplasmic reticulum, which stores a bunch of calcium. Now, as you may notice, the sarcoplasmic reticulum kind of sounds like the endoplasmic reticulum. That's because it actually is the endoplasmic reticulum. It's just a modified version of the smooth endoplasmic reticulum that we're all familiar with the organelles in the cell. But in the sarcomere, it's just been modified in order to store a bunch of calcium and maintain homeostasis of the calcium. So after this T tubule delivers this action potential down to these voltage sensors that basically block a bunch of calcium in to the sarcoplasmic reticulum. The sarcoplasmic reticulum is going to open these gates, and there's going to be a massive flood of calcium. This is why calcium is so important in your body. This is why you need to get enough calcium in your body, because it uses it for muscle contractions. It's going to be very hard to contract your muscles if you don't have enough calcium. It's going to open these gates of calcium, and they're just going to flood out all over the sarcomere, and the main role of this calcium is going to be binding to the troponin we discussed earlier. It's going to bind to the troponin, and the tropomyosin is going to shift, which is going to expose these little binding sites that the golf club shaped myosin is going to bind to. So these myosin heads that have been energized by ATP are going to attach themselves to the actin, to the binding sites that we uncovered earlier using the calcium. This is going to form something called the cross bridge. And after this cross bridge forms, the myosin head is going to pivot. It's going to pull the actin towards the center of the sarcomere. So this is going to make the Z lines move closer together as this happens. ATP is going to bind to myosin again. It's going to detach, and the head is going to reset again. It's going to go back to the position it was in earlier, and then it's just going to repeat the cycle over and over again. ATP is going to attach, its myosin is going to bind, it's going to let go, it's going to cock back, it's going to move the actin. Throughout this whole time, ATP is going to be attaching to the myosin. It's going to be going through hydrolysis, which is going to create energy by separating those phosphate bonds, and then it's going to detach in order to have the myosin move back and forth in order to move the actin. Okay, so after the action potential ends at the motor neuron, the acetylcholine is just going to be left there, and this enzyme called acetylcholine esterase is going to come and break it down. And then after that, the calcium pumps in the sarcomere are going to actively pump calcium back into the sarcoplasmic reticulum, so it's going to go back and get stored in there. And the troponin is going to release the calcium. The tropomyosin is going to block the binding sites again, so the myosin isn't just going crazy and doing the contracting. Now, if you have really high calcium in your muscles, there's going to be a risk of cramps, as the myosin is just going to keep contracting and it's not going to stop contracting because the calcium is just there uncovering the binding sites. So now, after going over all of this, we're going to go over some practical applications and how it actually relates to our life. First up is exercise fatigue. So why do we get tired at all when we're doing exercise? This is mainly due to ATP running low. Without enough ATP and energy being able to keep the myosin heads detaching from the actin, it's going to keep binding to the actin, and it's going to cause stiffness and reduced force. There's also just going to be a buildup of a lot of the metabolites that we used in order to do the contraction. There's going to be a lot of lactic acid and inorganic phosphate that are just going to be flying around everywhere. They're going to interfere with the cross-bridge cycling, and it's going to reduce the calcium sensitivity on the troponin. Also, it's going to start hurting a lot because the central nervous system is realizing, hey, we're kind of messing up our muscles. Let's send some signals to make this person stop overusing their muscle and potentially destroying the machinery in the sarcomere. Now, another really popular topic that connects to this is Botox. A lot of people get Botox in order to get rid of their wrinkles. They want to look better. But this Botox is actually botulism toxin. It's actually a toxin produced by botulism clostridium. Now, this toxin goes to the motor neuron terminal and stops the release of acetylcholine, which we discussed has to bind to the motor end plate in order to allow for an action potential, in order to allow for contraction. It actually binds to this neuron terminal and stops the acetylcholine from being released. Without this acetylcholine, the muscle fiber is just not going to be able to contract, and that's going to make the muscles stop contracting and reduce the wrinkles in the area that the toxin is in. Eventually, over time, there's going to be new nerve endings that grow, so they're going to have to repeat the whole process and put the toxin back in order to inhibit these new neurons. Another one we can look at is myasthenia gravis, which is Latin for grave muscle weakness. This is an autoimmune disease in which the body uses its immune system in order to destroy and attack a bunch of the acetylcholine receptors that are on the motor end plate. Now, with less of these receptors, acetylcholine can't bind to the motor end plate and can't produce an action potential, thereby causing less contraction and muscle weakness. One of the treatments is obviously to suppress the immune system. If the immune system is attacking your body and it's not doing anything helpful to your body, it's actually causing more harm, you would want to suppress it. A lot of autoimmune conditions are treated like this. They're just going to suppress the immune system. Another way to treat this condition is use medications to inhibit the function of acetylcholine esterase. That's the enzyme that breaks down acetylcholine. So by breaking down this enzyme, there's a higher chance of acetylcholine binding to the motor end plate receptors. And then the last one is rigor mortis. This is the condition that you see in people that have died. Usually 2 to 6 hours after their death, you're going to see that they're very stiff. Their muscles are going to be very stiff. They're going to tense their muscles a lot, even though they're dead and they don't have any conscious voluntary contractions. This is because when the breathing and the circulation of the blood stops, there's no more ATP being produced. So the calcium slowly leaks from the sarcoplasmic reticulum, and it allows the cross bridges to just form all the time. And without ATP, the myosin heads can't actually detach from the actin after they do the power stroke and move the actin. So they're just constantly pushing, they're just constantly contracted, and this is what causes the rigor mortis that we see in a bunch of dead people. But that's pretty much it for today's video. That's the sliding filament theory and how muscle contractions work. Let me know if you guys want me to go over anything else specifically. I'll definitely make some future videos about cardiac muscle and smooth muscle because they are just as interesting as these skeletal voluntary muscles. If you made it to the end of this video, be sure to subscribe and like the video, and let me know if you have any other video ideas that you want me to go over.